much welcomed and appreciated. Now I think I can introduce today's speaker, Karen van der Walt, Conservation and Science Advisor to Wellington City Council's Aotearoa Native Botanic Garden. And I'm just going to pass the screen to you so you can accept the invitation to present. Now, Karen has been immersed in the world of seed banking for several years now, and with a recent focus on seed banking for Myrtaceae species, her findings are relevant to what we might do in the face of the myrtle rust threat. Karen, your screen's looking pretty good to me, so I think you can get underway.
Thank you, Renee, and thank you for handing it over to me. Kia ora, everybody. Thank you very much for taking some time out to listen to me today, telling you a little bit more about freezing for the future and whether that can help us in our race against myrtle rust for our Myrtaceae species. So just a quick overview of what the talk will be dealing with. I'm going to have a quick snapshot about myrtle rust. There's been some beautiful presentations previously on it, so I won't delve too much into it. I will go down a journey about seed banking and what it is, what does it mean, what does it mean when we talk about desiccation tolerance for our seeds, and how does this apply to our native myrtles. And in particular, I'm going to talk about Lophomyrtus bullata, which is ramarama, Lophomyrtus obcordata, which is rohutu, Neomyrtus pedunculata, which is also rohutu, and then Syzygium maire, which is swamp maire. So without further ado, a quick journey down the myrtle rust route. So first of all, what happened to myrtle rust in Australia? Now, in some way, we're a little bit fortunate in New Zealand in that we can look at what happened in Australia since they've had myrtle rust since 2010. And up to now, they've got about 360 taxa that is susceptible to myrtle rust. And of those 360 species, 45 species are in decline. Directly due to myrtle rust, not necessarily other reasons. And I want to discuss a little bit two species in particular, and those are also the species that are in the photos that you can see. And these species are Rhodmania rubescens and Rhodomyrtus sissioides, which is the native guava. Now, neither of these species were threatened before 2010, so before the arrival of myrtle rust, these species were doing quite well. Now, both species are listed as critically endangered and likely to go extinct in the near future. And because they are so important to the ecosystem, it will cause a significant disruption as well. And it's been highlighted by Karen Somerville in her paper in 2010 that it is really important that ex situ conservation is done before we see significant impact on our species, especially for those species that we know are highly susceptible to myrtle rust. So if we take our journey in myrtle rust now into Aotearoa, into New Zealand, it was first recorded on Raoul Island in April 2017, and shortly after that in Kerikeri and North Island. And it is now subsequently established throughout the climatically suitable areas throughout New Zealand, and that was from 2020. Now, I can tell you a lot about what myrtle rust has done. There's been some papers published on it as well, but Graham Atkins, that's featured in this video, has really done the most beautiful video that was released last week on the rapid devastation of ramarama in the East Cape. And although we won't be able to watch the video right now because Renee sort of gave me strict orders that I can't go on talking the whole day, I really encourage you all to have a look at this video and kapai to Graham as well for the beautiful korero he did on ramarama, highlighting what is happening to our species. In addition to Graham's korero, what we also know is that if you look at the photos below of Syzygium maire, so that's the photo in the corner here, and credit to Rob Beresford for it. Those are myrtle rust spores that you can see on the floral buds of Syzygium maire, and that photo was taken in November last year, so it's a year ago, and that was from a population in Auckland. And that's really sad because those flowers are obviously what's going to become the fruits the next year, and those flowers won't be able to grow. And then the next two photos sort of supports Graham's video at the top, in which we have ramarama, which is Lophomyrtus bullata. This is a photo that I took of a plant up on the Capri Coast here in Wellington region. That was in 2019, so the red circle is around the tree. And then in 2021, this year, you can see that tree is basically dead. It's also in 2019, we collected a huge amount of fruit from that particular tree. In 2021, it failed to produce any fruits. So this is all quite gloomy, and I'm sure a lot of people are fairly depressed by now. But actually, there are some positives in what we can do against myrtle rust. Although it's a airborne pathogen, which makes it quite difficult to control, one thing we can do is we can look into seed banking. Now, what exactly we mean by seed banking is seed banking is when we store the seeds of any species to give us opportunities in the future to restore a species, to recover a species, maybe if it's gone extinct, and also to restore, enhance ecosystems. It's also a principle that's used for a lot of our crop and food species that we eat today are banked in conventional seed banks globally. So if you look at the picture at the top, that is Svalbard, which is in Norway, between the North Pole and Norway. And that's where a lot of our food species are banked. And as I said, that's using a principle called conventional seed banking, in which the seeds are dried to 3 to 7% moisture content, and then it's stored at minus 20 degrees Celsius. But I need to point out that seed storage is not a new concept. It's been done for many, many thousands of years. It started in ancient Rome. The Egyptians stored seed as well. What we really want here is the long-term survival. So we are ideally looking at hundreds or thousands of years that we can look after our seeds while they're in storage. When we do seed banking, it does involve quite a lot of steps. And this starts with the first step is that we collect good quality seeds and we collect them at the right stage of the seed development. So if we collect the seed too early, it might be able to grow, but actually it doesn't have all the reserves and everything else to look after itself when we put it into storage. Also, when we collect seeds, if we've got, let's say we collected 1,000 seeds, we want to know how many of our seeds are viable. So how many of them have got the potential to grow into seedlings and obviously big plants in the end. And that's really important because you will very seldom have 100% of your seeds that are viable. Now, the way in which we can determine if a seed is viable is we can first of all use a chemical stain called tetrazolium staining, in which we stain a seed to see if there's live cells in it. The second test, which is slightly better, is called a germination test. And that's, as it says, you grow the seed. Can we actually grow it? And then how does that seedling develop? And this is what we call the control or the baseline information or the benchmark. So everything that we continue doing to the seed from here on, we will always refer back to that initial information that we collected. And here are some photos. I always find photos easier to process than words. So you can see at the bottom, at the left-hand side, my left-hand side, there's clean seeds of, that is a Neomyrtus seed. And then the two top photos is TZ stain that was used on seed. So as I said, when you use the stain, if the stain stains your seeds red, it means they are viable and there's live cells. If they do not stain red or a pattern of red, then it means there's problem. And then you can see the one on the right hand side of that hasn't stained at all, which means that seed is no longer viable. But of course, that is where this test stops. So we can't say how well does our seedling grow, how good is it, how strong is it. So that's why a germination test is often preferred. And you can see the two bottom photos, that is also Neomyrtus, and that's a Neomyrtus seed starting to germinate, and that is actually on soil. And the seedlings are also Neomyrtus seedlings as they develop, and that was over a period of 8 weeks. So it gives us a really nice full picture of what's going on. So now that we've done the seed collection and we know how many is viable, we've got our control, the next thing we need to know is our seeds desiccation tolerant? And what that means is, can we dry the seed? So if you think about putting something with a lot of moisture or water into the freezer, it forms ice crystals, and those ice crystals will destroy the seeds. So we need to remove that extra moisture and prevent any ice crystals from forming. Now, if we're working with a species that we don't know anything about, we'll actually only do it on a small subset of seeds so that we know if we kill them, we didn't kill our whole collection. And what we do is you dry that seed, you can dry it over silica gel or you can dry it over saturated salts like lithium chloride, down to a low moisture content. You confirm that you've reached the right moisture content. And then really important, what you do is you do another viability test. So remember when we started with our fresh seeds, when we collected them, we need to do the same, and you need to do it in exactly the same way. So when you grew your seeds, you germinated your seeds, what temperature, what soil, what conditions did you use? So then we compare it. Now, if we find that nothing happened, so there's our pictures, our control photo, you can see there's a lot of healthy seedlings growing there. Then we desiccated the seed. So that seed's sitting in a desiccator that's got silica gel at the bottom. And then we do another germination test to see, first of all, do we have the same amount of seed that still germinate? And are those seedlings looking sort of similar to the controls that we haven't done anything about? If all looks good, then we proceed to the next step. We freeze a small sample. So still we're not working with a whole set, we're just working with a small subset. It's dried seed that we now put into a freezer or into liquid nitrogen if you want, or at 5 degrees. And after a certain amount of time, you do another test to see when you froze them, did they die, or do they still can look the same as to your benchmark or your control. So when we have all that information, what we can start forming is an idea of what our seed category is. So it's not really a category, it's more a linear system. So seeds don't fall into one of the three. It usually falls somewhere along the line of the three. So if we have those seeds that we dried and then froze and they still look the same as our fresh seed or our control, then we are possibly looking at something like an orthodox seed. So that's like we did with the crop species. And they are tolerant and they respond well to it, which is really good. If we have a problem, so let's say when we dried them and then we grew them again and we lost quite a lot of the seed viability, or maybe they were okay after drying, but they didn't like the freezing so very much, then we could be looking at something that sits along the intermediate line. So they're not quite comfortable, but they're not quite recalcitrant. And recalcitrant being species that really do not like being dried down. So the moment you lower the moisture content, you lose a lot of viability and they don't respond well. There's also some other clues that can tell us that seeds might not like drying. Big fleshy fruits sometimes do not like it. If a seed has got a very big embryo, a thin seed coat, and then generally seeds from alpine areas have been found to not tolerate the desiccation so well. Wetlands, which makes a lot of sense. If something dries in a wetlands, it likes to be moist. Rainforests, and then climate. If you've got a climate that gets really hot and dry, the seeds are probably going to respond better to dry conditions that we use for seed banking. Now let's continue our journey on seed banking and start looking at our Myrtaceae and what does it mean for our New Zealand Myrtaceae. So we've got 6 native genera, so I'm only referring to the native species here. And we can more or less group them into 2 groups. So the first group have got fruits that are really dry, fruits and seeds, and that's Kanuka and Manuka and Rata species. And you can see the photos from there. They form the capsules and they usually produce so many seeds, and then they are quite dry. And then the other group is the group that we'll be talking about more for the rest of this conversation, and that's the fleshy fruit. And those are the Lophomyrtus species, so ramarama and ruhutu, Neomyrtus, and then also Syzygium maire. Now I would like to introduce us to Lophomyrtus. So we're looking at Lophomyrtus bullata and Lophomyrtus obcordata. And what's really special about Lophomyrtus is that the whole genus only occurs in New Zealand. So nobody else has got Lophomyrtus. It's only us. It belongs to New Zealand. It is, once again, a really important species in the forest understory. And they produce fleshy fruits. And these fruits mature between May and June, which is actually quite a harsh time as well in New Zealand for animal species. They struggle to find food, so it's a good food source. And also where the 2 species co-occur, they form a natural hybrid as well. So sometimes referred to as Lophomyrtus ralphii, or sometimes just referred to as the hybrid between the 2. And you can see the photos, ramarama, and the 2 photos below each species of ramarama and ruhutu. You can actually see that the seeds look very similar. And when you cut them through, that white part over there is the embryo. And you can see the embryos look really similar as well. So what is our seed characteristics for the 2 Lophomyrtus species? So I just want to take a second to make sure, because even I still get confused. When we talk about fruit, this is the fruit. So it's the fleshy part that gets released. And within the fruit, you find the seed. These are clean seeds. And if we look at the 2 species, so Lophomyrtus bullata at the top and obcordata below that, we can see that bullata has got quite bigger seeds. They're heavier, but also they've got slightly more, there's more water when they are released, so they're more moist. We investigated the seed composition as well of Lophomyrtus, and the reason for that, so when you go to the supermarket and you look, oh, should I eat this chocolate bar, and you look at the back at what the ingredients are, and oh, it's got too much sugar, oh no, look at the fat, I'm not going to eat that. That's the same principle we used here. But what we are particularly concerned about here is fat, which I guess we're concerned about fat too when we go to the supermarket. But for seed storage, fat could give us a really important clue into what is going to happen to the seed. So you can see I encircled fat there. Lophomyrtus bullata, so ramarama, has got a little bit more fat than obcordata, rohutu, but neither species have actually got a huge amount of fat. So oily species will often have 30 or 40% fat in them. Now, the results I'm going to present to you now is based on 4 collections of Lophomyrtus bullata and also 4 of Lophomyrtus obcordata and 1 of the natural hybrid that we collected from the Skyline in Wellington. And the seed collections were made over a period of 3 to 4 years, so starting in 2018 till this year, 2021. And for the data collected, once again, we did the staining test. So remember that beautiful chemical stain that we can use, and then correlated that to a germination test. And you can see the seed germinating here at the bottom from ramarama. And then seedling health, which is really important, so that's seedling vigor. And the seedlings you can see in the photo down here are seedlings after 8 weeks in growing. I can see how big are they, so how tall are they, and how many leaves did they make, and how strong is their root system within that. I collected this for the fresh seed. So right in the beginning, like I said, we really need to know that. Then did exactly the same tests again for seed that's been dried. And then for seed that's been stored at 5 degrees, -18 and -196. So -196 is in liquid nitrogen. And that was stored over different periods of time. So from 14 days up to 3 years. I also want to introduce you quickly now to Neomyrtus before we look at the results, pedunculata. So Neomyrtus is slightly wider distribution. It's North and South Island and also on Stewart Islands. It's also really important in the understory. The fruit matures slightly earlier, which is in March, and the seed was collected from one population only from this year. So you can see quite a smaller data set, but it is still valuable to let us know what's going on. And with Neomyrtus, I collected the fresh viability, desiccation, and freezing sensitivity. But a little bit of a challenge with Neomyrtus was that the seeds are dormant. And what that means is when you expose those seeds to germination conditions, so nice warm temperatures with moisture and soil and everything it needs, it doesn't actually want to germinate. And that's nature's way of saying to the seed, hey, dude, hang on. The conditions aren't quite right for you. We want you to wait a little bit before you grow because the seedling won't have a good chance of surviving. And for us, it's sometimes difficult to figure out what the dormancy breaking cues are. It could be a low temperature, high temperature, a combination, or just time that's needed. So for Neomyrtus, I relied a lot more on the staining. So with the staining, you can see the 4 photos at the bottom. We've got a viable seed, that's beautiful red stain. We've got 2 partial stains where the one is part of the embryo is actually a little bit white and then dark and pink, which makes it difficult for interpretation, but then also very clearly a non-viable embryo that didn't stain at all. So this is where research always gets a little bit depressing because I've basically showing you 4 years of my life in one graph. But here you go. There's the 4 years of my life in one graph for Lophomyrtus, bullata, obcordata, also for the hybrid, and then there's 14 days for Neomyrtus. Now, what is important here is that we want to look at the control. Now, remember the control was the fresh seeds that we germinated. We didn't do anything to it. And you can see it was pretty good for all species. Neomyrtus slightly less, all above 80%. Now, when we dried that seed, what is important here is to notice, so this one is ramarama, the blue one. Look how much viability we lost when we started drying it. And remember, that's not what we want to see. When we looked at Lophomyrtus obcordata, which is rohutu, that desiccation did not affect them at all. In fact, it looks like it's almost exactly the same. Also for the hybrid between the two, which is super interesting. So if we look at, it's got bullata and obcordata genes in it, we can see that it actually falls between the two. So it's not quite as sensitive as bullata, but more so than obcordata. And then Neomyrtus didn't like it, even less than bullata. Now, the next 3 groups of bar graphs are the seeds that's been stored for 14 days only. And what we want to know here is Is there indication that our seeds are freezing sensitive? Do they not like it below 18? And you can see for that, nothing changed considerably. It was more or less the same after 14 days. However, when we looked at that for a period of longer than 1 year, so that is data grouped from 12 months to 36 months, what we have seen is not really good. So if we look at Lophomyrtus bullata, now we've had our control, which was really happy seeds at, looks like 90%, down to about 47%, stayed at 47 during our 14 days. It didn't mind being dried and stayed at 5 degrees for 12 months. But when we looked at what happens below freezing, we lost a lot of viability. It was down to less than 10%. And also for liquid nitrogen, although a little bit better, it's still not great. It's lost a lot of viability. Similar results for Ruhutu as well. So although they were very happy at 5 degrees, they did not lose any more viability. The moment they were below 18, things weren't going so well anymore. So we lost some viability, not as bad as a ramarama, but still too much to be comfortable with. And then for liquid nitrogen, we didn't do it for Lophomyrtus obcordata, so that's why that wasn't filled out. And once again, actually the hybrid did slightly better in liquid nitrogen. So this is quite a good overview to show us what is happening in a year. And even 1 year or 2 years or 3 years in storage is not a really long time for seed banking. Remember, we want to look at hundreds or thousands of years before we do that. Now, when we started wondering why is this happening, there's so many variables that could influence why seeds do not like it, or why do they die or lose viability. One really powerful machine that we can use is a machine called DSC. And what the DSC does is it enables us to do a thermal analysis of the lipids and the water as they go through the temperatures associated with seed banking. So from 25 degrees Celsius down to minus 100. And here's the graph that we get from the DSC. So you can see the cooling. There's the arrow. So this is our seeds. This is Lophomyrtus obcordata, Neomyrtus, and ramarama. And this is us, they going into their seed storage journey now. So they've been dried and off they go. And you can see this is them going through. This is 10 degrees. This is minus 10, minus 13. You get the idea going on towards the left. LC, remember those lipids that I referred to in the beginning, the fats. So we call fats lipids too. This is LC, it's lipid crystallization. So the lipids are forming crystals here, here, and here. And if we look at encircling, the range of which these crystals formed was minus 18 degrees to minus 28 degrees for Lophomyrtus bullata, where for the other two species was very similar. So it's minus 23 to minus 52. Now, if you quickly think back to what was our storage temperature for conventional seed banking, it's around minus 18 to minus 20. So there goes the red flag. We've got possibly a problem because we've got lipids forming. Now, if we look at the warming, so what's happened, our seeds are now sitting on this side. Remember, we cooled them down and they went on their cooling journey. Now they've been sitting here in DSC time only a couple of minutes, and then they start warming. So you can see here we're going now from left to right. And again, let's follow what's happening. LM, those are those darn lipids again. And what they're doing now is those crystals that formed, they are now melting. And the melting is what's giving us a hint into what's going on. And you can see for all three species, there are 3 lipid melt events. And once again, the last lipid, the second middle lipid event, melt event for bullata is again going very close to our seed banking temperature. So what does this mean? I think you all go, so what? What does this mean? Okay, for our study, remember I worked on 4 collections for bullata. They did not survive storage at minus 18, very low survival, and that's probably due to that lipid phase. So when we see those lipids crystallizing, it's not good for seed storage. Short to medium storage was definitely possible at 5 degrees Celsius. I have to point out that the seeds have to be desiccated, they have to be dried. I did the same experiment by not drying the seeds, so just collect them and clean them and put them in the fridge. They didn't even last 6 months, then they all died. So it is important to remove some of the moisture. Now, this is showing us there's problems for ramarama. It doesn't mean it's the end of the world, it's all doom and gloom. There's a lot more that we can investigate a little bit further. First of all, maybe if we use different cooling rates, those lipids will form in a different way that's not destructive. Also, this data is based on 4 collections only. Maybe there are collections out there that's got different lipid profiles that behave differently, that maybe are better for desiccation. Also, you can store seed outside of the lipid range, which is probably what we see why they survive 5 degrees better. Although we want to go below 0 degrees for longer term storage, maybe -10, or going into cryopreservation using a different method. And then, of course, we can also use a process of cryopreservation of the shoot tips, which is what the photos show at the bottom. Now, just quickly, a snapshot for Rahuto and Pendiculata. Once again, with Rahuto, the seeds were not desiccation sensitive, but there was a lot of variability. So one of the populations were okay, they actually survived, 63% of the seeds remained viable, while the rest of the seeds died. So there is 2 populations that didn't like it and 1 was fine, and we're not quite sure why. It could be the lipids that wasn't stable from the one. Once again, 5 degrees was really good for short-term storage. And as with ramarama, more research into whether this is consistent throughout the populations. Also, for Neomyrtus, to point out, it was a short-term study in terms of seed banking, but the seeds are desiccation sensitive, and they are likely not to like -18 if we look at the DSC results, and there's also the dormancy. So just for me to wrap up quickly, I'm going to talk about Syzygium maire. So this is a refresher of where we've been. We talked about Lophomyrtus, we talked about Neomyrtus, and we're quickly going to touch on Syzygium maire, which is our last fleshy species. So Syzygium maire, it is a swamp species. It's a swamp maire. It's called maire tawake. Once again, belongs only to New Zealand. It doesn't occur anywhere else. It's confined largely to the North Island, where it grows in the swampy areas, and populations have declined by 70% even prior to myrtle rust. And as you remember from that photo, myrtle rust is having quite an impact on them. Now, because the swamp maire fruits are quite big, we need to focus here on working on the embryos. So just like we worked on the seeds, remember the fruit versus the seed, here we work on the embryo, and you can see the embryo within the seed, and then the embryo gets germinated and grown in vitro in culture. So you can see in the petri dish at the top. Just like we did with the seeds, we need to determine whether those embryos are desiccation sensitive. And we do that through the same process. So we dry them, this time in a laminar flow cabinet. And this is the survival of the embryos, the pink graphs, and then the red line is the moisture content. And for swamp maire, this means that it is highly recalcitrant. So you can see how quickly the viability drop off even before we get close to seed banking moisture contents. If you want to read more, you can refer to the reference in this slide as well. So for swamp maire, the only option we have is cryopreservation, and that's storing in liquid nitrogen. And the cryopreservation process does take quite a few steps, and each step needs to be optimized. So when we take out that embryo and we grow them, we need to make sure we don't hurt them. We give it the right food and nourishment to grow. How do we sterilize it so it doesn't get sick while it's in vitro? How do we cryoprotect it? So even though it doesn't like drying, we have to remove that moisture because ice crystals are bad. And how can we do that? Freezing them really quickly, thawing them really quickly, and then for each one, for swamp maire, figuring out what can we give them to make them recover after this process and grow into normal seedlings. So with the swamp maire, we've tested various protocols. The first one, as you can see, the embryos there on the desiccator. So that's direct cryopreservation. They get dried a little bit, but very quickly, and then put into cryovials and plunged into liquid nitrogen. The next step we tested was encapsulation, dehydration, cryopreservation. And what that means is that in there is a little embryo, and that is an embryo encapsulated in a little bead. And that bead gets dried a little bit as above, and then put into liquid nitrogen as well. And then also droplet vitrification, which basically means it's antifreeze for plants. So it does remove the moisture, it puts a chemical in instead of the moisture, and that chemical will not freeze. And then we use droplet vitrification. So that's a little aluminium foil with an embryo on top. Now, once again, Renee said to me, you cannot go on the whole day. People have got to return to their jobs. So unfortunately, I cannot go into too much detail with the results from the cryopreservation, but the results have been published in various papers already. But the summary for swamp maire is that the embryos are highly sensitive to desiccation. We also did the DSC scan for swamp maire, and we saw that the embryos need to be dried to below 0.2 gram per gram moisture content to prevent ice crystals, but this is lethal to the embryos as well. So we can investigate further by increasing cooling and warming rates for swamp maire, and then also adding antioxidants to it. So in conclusion, seed banking is a really powerful tool for us against the race against myrtle rust. But we need to be really careful when it comes to our fleshy Myrtaceae species. Lophomyrtus did not survive storage at -18 or -196 in the study. Although they retain better viability at 5 degrees Celsius, storing at 5 degrees Celsius is not a long-term seed banking option, as it's likely to lose viability within 10 years. Similar results for obcordata. They were not desiccation sensitive, but they did not really like -18. This could be due to the lipids. It could be due to variables associated with different populations. It warrants further investigation. With Neomyrtus, they are desiccation sensitive, and based on our DSC results being that they are likely to continue losing viability at -18. Swamp maire will require the ongoing research to optimize the cryopreservation of the embryos. However, we can also look at shoots and plumule as well. So my final message is that myrtle rust has already had a significant impact on Lophomyrtus bullata, and the fruit production has been severely reduced. We really need to urgently investigate more suitable storage options for the 4 fleshy species in New Zealand to ensure that these beautiful taonga remains part of Aotearoa. My acknowledgements, it's always a little bit difficult because there's a lot of people, there's projects or collaborations, but I do want to acknowledge the Line Club of Karori for funding for the lab. Also to Narangingi Iwi for allowing access to the takiwa and the seed that was collected, and also seed collection assistance from James Jones and Jennifer Alderton Moss, and also to all the access that was allowed by landowners. If we don't have seed, we can't work on it. And funding was provided by MB and MPI as well. Thank you very much for taking time to listen to me today, and I hope you enjoyed my talk.
Thank you very much, Karen, for that very extensive presentation and excellent education for those of us that are not familiar with seed banking. I don't see any questions in the chat box yet, so hopefully that's not a technical problem on my part, and just have a bit of a chat then and see if any come through. So how unusual are these results in terms of the challenges they're presenting for seed banking in the context of other species, maybe crop species versus native species? I mean, are there things which are just really straightforward, and is that common?
Yes, that's a good question. Thank you. Obviously, I've presented on the difficult species, so our problem children I've presented. But yes, conventional seed banking actually is considered to be suitable for close to 90% of our seed producing plants in the world. And that includes, remember our slide when we brought up the dry Myrtaceae species and the fleshy ones. For our dry Myrtaceae, so Manuka and Kanuka and Rata species, seed banking will work. So that's our easiest step of just banking them at -18. So definitely it's suitable for the majority of species, but we do need to have data and ongoing research into this.
And speaking of which, we do have a question in about this, being what are the next steps for your research? That's such a broad question, it could be a little tricky. Perhaps we could interpret that as, you know, if you had to prioritize 1 or 2 things to do next and you were able to do those, what would they be?
Ramarama would be a very big priority to understand what we're seeing here at -18. How does that apply to the whole population, or at least to additional collections, maybe collections from further up north or east coast or somewhere like that? And if we continue to see that problem, what can we do? Because Ramarama is running out of time. So it's really urgent to just understand what's going on with Ramarama.
Absolutely, and I guess the flip side of the coin is, I'm not sure if you are able, perhaps happy to talk about this at the moment, but what, you know, are there things you're able to do further at this time? Is funding out there for you to carry on to any extent?
Yes, so definitely as I said from the beginning with the first slide where you've got seed collection. So seed collection needs to be prioritized. If you don't have the seeds, you can't collect it. And as we've seen, at least you can buy time by storing the seeds at 5 degrees. So collecting the seed, collecting them from different populations, having a look at whether those lipids are the same, the profiles, even that, because we went into more details with the fatty acid profile. So what kind of fats are in there? Is it similar for all the populations? And then if we store them, as I say, if we can dry them slower, or sorry, freeze them slower, they might be able to survive that. So um, I think it's not a question. If we want to continue, we have to continue to make sure, because we do not want to put all our resources into seed banking at -18 for ramarama specifically, if it's not the right way to go. So verifying and confirming that this is applicable to wider collections as well. So it's really important.
Very good. All right, the questions are flowing in now, so let me see if I can pick some of these up. Lots of thanking you very much and mentioning that was a really well-structured, extensive presentation. Have you tried treating Lophomyrtus seeds with cryoprotectants to improve survival in liquid nitrogen, something that we're considering for a few Australian species?
Yeah, thank you for that. That would be one of the next steps. So what I didn't present here is that for ramarama, we also looked at not drying them quite as low. So remember, they lost a lot of viability when we desiccated them. So we went one desiccation moisture content level higher, and then this DSC scan showed us that there is a lot of ice crystals. So yes, the next step would be to look at using cryoprotectants. So the antifreeze would be the next one. So it's prioritizing what we're doing. So the first, now we sort of saw, okay, there's trouble here for Lophomyrtus in what we're seeing. Confirmed, we saw it. Our scientists, we have to confirm many times that we're seeing the right thing. And yes, the next step now is, hey, what are we going to do? Can we use cryoprotectants? Can we do cryopreservation, slower cooling rates? So yeah, thank you for that. That is a good next step to investigate.
And on the theme of cryoprotectants, what cryoprotectant do you use? Something that penetrates the cells, such as glycerol?
Yes, so we use PVS2. That was for swamp Myrtle specifically. So we've only tested PVS2 on swamp Myrtle, and the work, what we focused on is using PVS2, but modifying the application method. So conventional or VIV, so vacuum infiltration vitrification.
Very good. And I'm sure audience members, if you had further technical questions for Karen, she'd be very happy to address those via email, and I can pass those on to her as well if you do want to get in touch. A little bit of a new direction are the seed microbiomes being explored. Within the Beyond Myrtle program, we haven't publicised this widely yet. We're going to be looking at seed microbiomes for the Aotearoa. I suspect that's not being done much elsewhere in New Zealand, and I wonder, Karen, is that an approach which has been explored overseas, assuming that you haven't looked at seed microbiomes?
It hasn't been explored, but it's been highlighted, if that makes any sense. So we're spending so much time in understanding what's happening to our seeds when we desiccate them and freeze them. But at the same time, acknowledging that there is a microbiome that can be beneficial to the seed as well, and what happens to them when we take them through the process. I guess the challenge here is we don't even know yet what that microbiome is. So we need to go back to sort of our fresh viability tests and say, okay, what is the microbiome? What does the microbiome look like when we take them through the steps? And do we destroy them when we take them through that step? So some of my germination stuff we've moved away from sterilizing the seed extensively and growing them in sterile conditions to actually growing them on soil, so that they don't need to be sterilized. And I have seen the seedlings appearing to be more robust and healthier. So maybe that is a microbiome, maybe it's sterilization, but I think that warrants a lot of further investigation in the future.
Certainly interesting to think about.
Yeah.
More on technique here. Could you snap freeze the growing part of the embryo in liquid nitrogen and later germinate the seed on a nurse endosperm, as has been done with clover embryos germinated on gorse?
Yes. So that is sort of what I glanced over, and I do apologize, but time restraints. So I have started looking maybe into the shoot tip cryopreservation. So that's a process where we grow the seeds, and with ramarama, what we have, we proliferate it. So you've got lots of different individuals, and then the same process that I follow with swamp Myrtle, we do it for those shoots. So that is the next step, and the good thing is that it is often easier to achieve than using a whole seed or a whole embryo. But the important thing here for ramarama is just, first of all, making sure conventional seed banking doesn't work, because that is the easiest way to do it. Now that we know it's not possibly the best way, we need to look at it and go down the journey of swamp miring and see if that's gonna be required for ramarama. And of course, keeping in mind it's very urgent to get it done.
Something a little bit different here, but an excellent question given our circumstances. How to guarantee that myrtle rust spores are not conserved with the seeds?
Yes, that's a very good question, and thank you for that. And what I think Rohan's been looking at, and hopefully Scion is continuing, is they are looking at whether the myrtle rust spores are present within the seeds. At the moment, some of our collections we've only made from areas that are myrtle rust free, except we are getting to a stage, especially for ramarama, where we will not have that option anymore. So that is not something I can say other than not having spores within the seeds, which we don't yet know. It's a consideration, but also we want to get to a point in seed banking where we've banked our seed and we know how to do it before we have to be desperate in our seed collections. So yeah,
That would
hopefully
certainly
that.
be the ideal scenario. So there's one more question in the chat box here, which is great because it's going to be a good one to wrap up with, and we probably should be wrapping up shortly. Given this dire situation, is there a way people can help support the work you do, such as helping collect seed from some of these locations that have not been tested yet? Which, and yeah, similar one here. Based on this work, what could concerned community members, school groups do to contribute to seed banking now? And this does loop back again a little bit to, are you carrying on with this work at the moment, and to what extent, and can people help you?
Yeah, so definitely. I think whatever we do, I don't want to, hopefully I didn't paint a too dire picture, because what I'm trying to say is, hey, don't give up. Don't think all's good. Let's just put a big effort into this. Absolutely. If there's seed, we need to investigate seed further. For collections is a good indication of what's going on. But if you might have different genes, different identities within your raw hair or your area for ramarama, maybe a stronger gene that responds better. So that is definitely yes, answering the question, more seed so we can investigate and confirm what we're seeing. And also, I've talked about seed banking, but let's not forget the importance of planting. So I know some people moved away, especially from ramarama, saying, oh, well, they're really susceptible to myrtle rust. Let's not give up. You know, there could be resistant genes within a population that we just don't know yet. And that's longer term. We might realize, hey, there's some that might be okay. So yeah, the community can be involved, seed banking. And also, once we have the good data on seed banking, we'll be able to help communities to seed bank in the right way that suits ramarama.
I
Yeah.
think the question there might be, though, in the immediate sense, are you interested personally in hearing from people that have
Yes.
seed that they would like to supply to you?
Yes, please. Definitely.
Excellent. This
Excellent.
is
Yes.
great news.
Yes.
And so I guess, yeah, we did not anticipate this, you and I, when we were chatting about,
Yeah.
and we haven't discussed yet how you might like those people to get in touch with you. Is this something
Um,
you've thought
email's
about?
fine. Yeah, email's fine, and we can take it from there and see
Okay.
when we collect the seed. I just want to also emphasize to people that please make sure that we have the right conversations before we collect seed and
Yes,
send it to
certainly.
us. Yeah, yeah. So I know
There are
people
mana
can get
whenua.
really excited and compassionate
Yeah.
about collecting seeds, and then maybe just offend people by collecting seed that shouldn't become. So yeah, drop me an email and we'll discuss it. But definitely we're interested in seed and we need to get the seed to see what's going to happen.
Certainly, there's real implications here for mana whenua for this kind of work, and we certainly
Yes.
want to ensure we're always following best cultural practices and working in partnership with iwi where appropriate. Excellent. Well, I'll look to provide your email in the follow-up email then. And of course, people can probably find that through the Aotearoa Native Botanic Garden, and they can certainly get in touch with me via our website. My email should be there, and the Beyond Myrtle Rust program. Excellent. Well, thank you very much. Again, let's wrap up that webinar for today. A video of today's webinar will be made available on the Beyond Myrtle Rust website. That usually takes a few days to make its way there. You'll probably see it there early next week. But everyone who registered for this webinar will receive a copy of the recording if we didn't have any technical problems in their inbox on Friday. Now, there will be one more webinar before Christmas. It will be Wednesday, December the 8th, and we will be hearing from Rod Fincham, Associate Professor at the University of Queensland. His talk title will be Unprecedented Extinction of Tree Species by Fungal Disease. So he's not holding back there. And that talk title is named for a research paper that he recently published titled as such. Now, this work does focus on threats to Australian Myrtaceae, but I think we will be pondering what the learnings might be from that for New Zealand. Until then, ka kite, and I'll see you later.